Good afternoon. It's great to see all of you here. Thank you for joining us today to celebrate the installation of Petra Levin as the George William and Irene Koechig Freiberg Professor of Biology. On behalf of Washington University and Arts and Sciences, I extend my warmest welcome to all of you attending in person or watching our live stream. We have a small change in today's program. Ram Dixit will not be able to join us today. Instead, I'm pleased to have Jonathan Lossos, the William H. Danforth Distinguished University Professor, to introduce Petra later in the program. We are delighted to have some of Petra's family members and special guests joining us for this wonderful occasion. They include her husband, Drew, daughter Isabel, and son Dwight, her mother Miriam, her friend Susie, and Susie's children Will and Sophie, present and former members of the Levin Lab, and Ralph Quattrino, Emeritus Dean of McKelvey Engineering, and Spencer T. Olin, Professor Emeritus of Biology, who recruited Petra to WashU many years ago. Will you please all stand? I can see that many people want to be Petra's special friend, so you can all stand again if you want. So the beginning of another stimulating academic year is upon us. I love the energy on campus during this time. It's always such a pleasure to welcome exceptional new students, including Petra's daughter, who is sitting right there, and stellar faculty to our campus and thrilled to embark on new courses, programs, and initiatives. There's no doubt that in arts and sciences, we're building on the tremendous momentum of the previous year. The initiatives from our strategic plan are fully engaged in meaningful transdisciplinary work. We have welcomed an unprecedented number of new faculty this year, a total of roughly 60. We are prepared to top last year's record-breaking research funding, and we are progressing on our exciting plans for a new signature building in the center of campus. This academic year also began with particularly exciting news about a milestone investment in our student community. Washington University has adopted a no-loan financial aid policy, and that is just so profound to me. I'm so excited. It's part of its sustained commitment to removing financial barriers for admitted undergraduate students. This policy will shape the arts and sciences and WashU community in profound and positive ways. This investment is part of WashU's bold vision for the future, one that complements our own transformative vision for arts and sciences. Our vision is ambitious. It pushes the boundaries between disciplines, departments, and programs. It emphasizes diversity of thinking and perspective, and innovation and tangible public impact. The scholarship of Petra Levin is an excellent example of this kind of work. She is an innovator, a collaborator, and a passionate advocate for access and inclusivity in the sciences. Her research focuses on how cells respond to changes in the environment by altering their growth, shape, and composition. This work will help us understand how human pathogens are so adept at acquiring antibiotic resistance, a growing crisis in today's world. In the United States alone, approximately 2 million people a year become infected with antibiotic-resistant bacteria. Excuse me. Petra is also one of the faculty leads in a new cross-campus research cluster focused on the development of imaging tools that enhance the study of infectious diseases. It's exciting work which brings together scholars from biology, physics, chemistry, and the School of Medicine has been funded by the new Incubator for Transdisciplinary Futures, a signature initiative of the Arts and Sciences New Strategic Plan. You will hear more about Petra's outstanding career and impact in just a few minutes. And now I would like to acknowledge the generosity and enduring legacy of George William and Irene Koechig Freiberg. Dr. Freiberg received his PhD in microbiology from Washington University in 1917. Upon completing his doctorate, he served in England and France during World War I as part of the university's Base 21 Hospital, a US military hospital staffed by doctors and nurses from the Washington University School of Medicine and civilian volunteers from St. Louis. Following the war, he found his way back to St. Louis to join Anheuser-Busch as coordinator of research and patent information. He retired in 1953. Irene Koechig Freiberg received bachelor's and master's degrees in chemistry from WashU in 1911 and 1912, respectively. And in 1919, she was one of the first women appointed to the faculty of the School of Medicine at WashU. For 30 years, she taught biological chemistry and microbiology at the Washington University School of Nursing and authored a number of scientific articles, as well as a textbook, Chemistry for Nurses, published in 1930. Dr. and Mrs. Freiberg were among the recipients of Washington University Alliance Appreciation Award, now known as the Robert S. Brookings Award, which is given to individuals who, by commitment and generosity, exemplify the alliance between WashU and its community. We are indeed grateful for their commitment. Through their state gift and 3 endowed professorships, Dr. and Mrs. Freiberg have ensured that our Department of Biology continues to recruit and retain stellar faculty like Petra. So now I would like to invite Jonathan Lossos to the podium to share more about Petra's outstanding scholarship and career at WashU.
Good afternoon, everyone. Ram Dixit, chair of the biology department, sends his regrets that he cannot be here this afternoon, but I was very pleased to be asked to fill in for him. In fact, it's a great honor to introduce Dr. Petra Levin as the George William and Irene Koechig Freiberg Professor of Biology. Now, Petra and I are kindred spirits. I love lizards, Petra loves microbes. Microbes, of course, are important as the first life form on Earth and as agents that deeply impact many biological and physical processes that are critical for the health of the planet and our own well-being. And so what's not to love? Petra's journey in microbiology started as a graduate student in Rich Losig's laboratory at Harvard University, where she used genetics and cytology to identify factors responsible for asymmetric septation at the onset of sporulation in the genetically tractable soil bacterium Bacillus subtilis. I suspect that many of you, like me, don't know what a number of those words mean. Hopefully Petra will tell us. Petra then carried her love of Bacillus subtilis onto her postdoctoral work in Alan Grossman's lab at MIT, and then subsequently, as she set up her own lab when she started as an assistant professor in the biology department here way back in 2001. Here at WashU, Petra expanded on her past work to identify factors that mediate the assembly and correct localization of the tubulin-like cell division protein called FTSZ. This protein forms a ring-like structure that defines the division site in bacteria. In a landmark 2007 paper in the journal Cell, Petra's lab identified a metabolic signaling network that provided the first molecular understanding of the relationship between nutrient availability and cell size. Focusing on the intersection of the environment and microbial physiology, Petra's lab continues to be at the forefront of discovering the mechanisms governing bacterial growth, cell cycle progression, and antibiotic sensitivity. While Bacillus subtilis continues to be a favorite model microbe in Petra's lab, her research now encompasses evolutionarily divergent microbes, including E. coli and the pathogen Staphylococcus aureus. Indeed, a defining feature of Petra's career is her willingness to use all means necessary, from divergent model organisms to diverse experimental approaches, to address fundamental biological questions that have broad applicability. Petra has a remarkable record of grant support, having been continuously funded by the NIH since 2002, and her lab has published over 70 research and review articles in that span. A few of her awards and honors include the prestigious NSF Career Award, which she received in 2005, a Fulbright U.S. Scholarship in 2015, and election as a Fellow of the American Academy of Microbiology in 2018. She is also a highly sought-after speaker at conferences and seminar series. Petra is committed to undergraduate and graduate education and has served as co-director of the Plant and Microbial Biosciences Graduate Program, which she helped establish in 2013. She taught the undergraduate microbiology course for 13 years and currently teaches a course on the history, pathology, and prevention of infectious diseases, a timely topic indeed. She is also a proponent of broadening access to research at all levels. In 2021, she developed Enter, a university-wide initiative designed to increase participation in research of low-income, first-generation undergraduate students. In sum, Petra is obviously a very worthy recipient of this honor. It's my great pleasure to introduce the George Williams and Irene Koechig Freiberg Professor of Biology, Petra Levin.
All right, are you ready for the big moment? It is my honor to present you, Professor Levin, with this medallion that signifies your appointment. On the front, it reads, George William and Irene Koechig Freiberg, Professor of Biology, Arts and Sciences. And on the back is inscribed, Petra Levin, September 19th, 2023. For the record, I knew all those words that Jonathan said, so I'm very excited for your lecture.
I'm shorter than everybody, I think. So it's really a pleasure to be here. It's really an honor that all of you came. And I really want to thank the Dean and the Provost for this honor, and also just everybody who's here. Sorry, I'm not going to cry. I do want to thank George and Irene Koechig Freiberg. I did a little research on them, and they were actually kind of amazing. But actually, the first Freiberg professor was Roy Curtis. And this is a picture of Roy when he was younger than when I met him in 2001. But he's actually a very famous microbiologist, and he was here at WashU with his wife, Josie Claire Curtis. And he had gray hair by the time I got here, and he would play Santa at the Christmas party, which was actually quite spectacular. But when he was gifted it, Dr. Koechig said that Mrs. Freiberg and I have remembered the excellent education we received at Washington University and the importance of great teachers in assuring future students of quality education in the sciences. And I really feel like that's our mission in biology, and I was really happy to read that. He also studied plant mosaic diseases. It was before, I think they knew that they called them viruses, but they hadn't really imaged these little things that cause the disease in plants. And he studied them, and he has a whole bunch of black and white images in this paper that's published in the Annals of the Missouri Botanical Garden in 1917. But for the life of me, the quality of the images was such I couldn't actually see any evidence of disease. I'll just have to trust him and say it's a poor copy. Irene Koechig was also kind of amazing. There aren't that many pictures of her. I sort of found this one in an old hatchet, which is what they called the WashU yearbook. And she was from St. Louis, and she entered as a transfer from Missouri University from Mizzou. And after she graduated, she went back to Mizzou for a little bit, and then she came back to WashU to teach in biological chemistry at the medical school. She taught nursing. And I think this is impressive and inspirational for anybody who's tried salary negotiations, because her first year, she only got $360, and she was also expected as her annual salary, and she was also expected to devote part of her time to working in metabolism ward in addition to chemical laboratory. I'm not sure what that means. She's not a medical doctor, so. But the following year, she must have been really good, because they raised her salary to $750, which is very impressive. And she wrote this book, The Chemistry for Nurses, which is lovely. The introduction is really nice and explains chemistry, and then has a handy dandy, for those of us who sometimes get confused between yards and meters and centimeters and inches, it has very handy equivalency, which I thought of printing out and posting for some of my students. I want to start by thanking my family who supported me through all of this, and I always feel like the tensest times are in my house are when I'm trying to finish a grant or I'm waiting to hear on a grant, and this time NIH really made it very difficult, because they went from scoring, it took 6 months to actually get funded. I'm not sure exactly why, and I thank Linda Hurt and the accounting office for keeping me sane. But I really want to thank my husband Drew, Isabel and Dwight, my son and daughter. And I also want to thank my extended family. My mother is here. She's in the red shirt. Her hair color's a little different in this big photo of my extended family. And my aunt, Margaret, who reminds us all to be silly sometimes. And then I also want to thank Susie Stark, my friend, who's here from OT, and her daughter Sophie, who's a freshman. Sophomore at Washington University. I'm never going to live that down. I do want to say my daughter Izzy is also a first year, no longer first year here, and is on the cross country team, so she represents WashU. Okay, so the science. I'm going to try not to use too many words besides Bacillus subtilis or E. coli. But what I really want to talk about is work that we've done in the past 15 years, and I really want to thank my lab, who is here, including Lizzie Muller and Corey Westfall, who are alums of the lab, as well as all the other members of my lab, and I will mention all of their work, and especially my undergrad, who's here, Nina Zhang. I want to talk now, though, about the work in the lab, sorry. So I think we're all familiar with the idea that genotype influences phenotype. I always try to squeeze in a picture of my dogs into talks. The big one is Lecker, the little one is Maymay. They're both dogs, but they obviously look very different, and that's because of their genes and their genetic makeup. As anyone knows, if you can look at your parents or your siblings, and you can also tell that you're related. I think one of my parents' friends said of this photo of us, and you can guess which one is me. It's probably around 1974, 75. You can tell also by the dress, but that we look like a family of clones. We are not clonal. But I do, when we think about more in my lab, we do think about genetics, but we think more about how the environment influences appearance. And we all know the environment influences appearance. One of my favorite examples of this is Fat Bear Week, which is coming up. It's the first week in October, and you can vote for your favorite giant grizzly bear. But grizzlies gain about 1 kilogram a day prior to hibernation. So this is the same bear, one fishing and then I guess waiting to go into their cave. I don't think they can move that much farther. But this bear is fishing for salmon and they just basically eat salmon all day. And somebody told me, and I don't know if this is true, but they just eat the brains of the salmon and then they toss the rest of it on the side of the river because it doesn't have enough calories, it's not worth it. So, sounds possible. But I want to convince you guys a little bit that bacteria are just like bears. If you grow them in medium, that's basically like iceberg lettuce. So it doesn't have very much, just enough for them to grow on. They tend to be smaller. These are actually E. coli, and thin and short. If you grow them in the equivalent of a Big Mac and an extra large soda, a supersized soda maybe, they are bigger, they're longer, and they are also wider. So in this way, bacteria are also influenced by their environment. These are genetically identical organisms. So remember, single-cell organisms, they don't have the advantage of a skin and multiple layers of cells. They just are bathed in whatever they're in. So the environment is to bacteria, it's everything. It influences how they grow, what they look like, and it influences how sensitive they are to antibiotics. And that's really kind of what my lab is interested in. Bacteriologists, though, we are creatures of habit. We tend to use the same medium over and over and over again. These are just 3 examples. It's not really important that you remember them, but they're just common medium. And if we use LB for one experiment, we use it for the other experiment because then we can compare them, but we don't vary it. And these are just 3 different types of medium. We usually pH them to neutral pH, again, just to make sure everything is the same. But obviously, the environment is not the same as those medium. These medium, they were selected long, long ago by scientists who wanted to have the bacteria grow as well as possible, as quickly as possible, and make as many more bacteria so they could study them. They are like a sunshiny day. But obviously, the real world is not always a sunshiny day, although lately in St. Louis, it seems that way. Sometimes, though, you really have to start thinking like your organism. So this says, day 37, I finally earned E. coli's trust. And I will convince you by the end of this that no way should E. coli trust any member of the Levin lab. But, so the outside of the bacteria, which we call the cell envelope, and it consists of a cell wall and a cell membrane, and sometimes an extra membrane and some other things, that is completely exposed to the environment. But it's a very important place because that's how the bacteria see the world. All the things they need to attach to things, sense things, move through the environment are stuck on the outside. So it's very important. It keeps the insides in, it keeps bad things out, it lets good things in, like sugar, for example, and it gives bacteria their shape. Bacteria come in many shapes. There are even some that are some archaea that are squares, round, semi-round. There's many, many shapes, spirals, and that's really the cell wall that gives them that shape. But if the outside of the cell wall has a problem, and this actually is a movie from Lizzie Muller, and they explode. So it needs to be kept intact. So one of our big questions has been, what happens to these things on the outside of the cell when the environment changes? What happens to the cell itself? How it grows, how it responds, what it looks like. And I'm going to tell you about work. I'm not going to tell you about too much cell division work. I'm really going to talk about how the environment impacts cell size, how both nutrients and also pH, and I'm going to talk a little bit about antibiotic resistance. And these are very short stories, and you don't have to really remember any of it. There is no exam. But why do we study this? As Dean Hu and Jonathan noted, there's a real problem right now. The CDC has antibiotic-resistant bacteria that essentially you can take and you'll get an infection, you'll go to the pharmacist, they'll prescribe you an antibiotic, you'll take it, and it won't do anything. So the bacteria have a way of evading that antibiotic. And this is a really, really big problem, and it's growing. So these data are the 2013 data, but when they updated them, so there are about 2 million illnesses and 23,000 deaths. So illnesses have gone up to 2.6 million, and there are 44,000, so the number of deaths has almost doubled. And actually, a really dear friend of mine who's a doctor, a pediatrician on the West Coast, almost died of a lung infection from Staphylococcus aureus, which 20 years ago would have been not a problem. He was basically really close to death. And now he's better, but it was a really scary time. So the bacteria that we work on, the bacteria that we've been focused on most recently are E. coli and Klebsiella pneumoniae. And both of these are leading causes of deaths from antimicrobial resistance. So when people die of a disease or they have an antimicrobial infection that's resistant to antibiotics, often it's E. coli, often as a bladder infection, or Klebsiella, which can cause bladder infections, or it can also cause lung infections and sometimes become systemic. And so we hope by understanding how the cells respond to the environment and how that also impacts directly in lab antibiotic sensitivity, we can at least understand and maybe come up ways to make our antibiotics that we have better. So I mentioned that nutrients make bacteria bigger. That was sort of our first foray into looking at the environment and bacterial growth and differentiation. So again, this is E. coli. They're about 3 times larger in volume in nutrient-rich medium, the Big Mac medium, than they are in nutrient-poor medium, the iceberg lettuce. So work that was done by a bunch of people in my lab, starting with my first grad student, Brad Weert, Visco Hill, and Jenny Chen, so 3 students did all of this work. They figured out the molecular basis of part of this increase in size. So they increase in width and they increase in length. And so the length increase turns out to be in large part because carbon-rich nutrients, so carbohydrates in the medium, are converted into a molecule called UDP glucose. You don't need to remember it, but probably glucose is familiar to you. It's a sugar. It binds to a protein, and this happens in Bacillus subtilis, and it happens in E. coli. And when the levels of UDP glucose inside the cell are high, which they are if they're growing in carbon-rich medium, in medium that has a lot of sugar or things with carbon in them, like carbohydrates, that protein actually interacts with the division machinery and kind of makes it very difficult for the cell to divide. So it can't divide, and it's growing longer and longer, but it can't divide. So the end result is that the cells are longer in the nutrient-rich medium. If they're growing in nutrient-poor medium, so the iceberg lettuce medium, there's very little UDP glucose, and that protein that would interact with the division machinery actually doesn't. It actually forms these beautiful hexamers with itself and doesn't touch the division machinery, so the division machinery can just do its job. And so the cells divide very easily, and they're shorter. And so there's a lot of beautiful work in this, but basically the idea is the cells sense the carbon that's in their medium as this molecule UDP glucose, and that acts to interact with the protein that then inhibits division to increase cell size. We also found, and this is work by Stephen Vidia, a postdoc in the lab, that nutrients influence the capacity of the cell membrane. So bacteria don't store, at least E. coli and Bacillus, don't store fat the way we do. The only lipids they really have are in that membrane that holds the cell, cytoplasm, and all the insides in. That membrane is made of fat or lipids. So if you grow cells in media with lots of carbon, they can make a really big membrane, and that cell can be much bigger. If you grow them in a medium without too much carbon, they actually need that carbon for other things. They need that carbon to make DNA, RNA, other things. So they make a much smaller membrane, and that also helps keep them small. So those 2 things, that's not the whole story. There are many other things that modulate size, but those are 2 ways that bacteria connect size to what's available in their medium. So we were doing this work, and I have this student who's in the audience, Lizzie Muller, and she did a control. So we do good controls. And she discovered when she did her control that actually if she takes the medium and she makes it more acidic, so more sour, the cells are short. If she makes it less acidic, E. coli is longer. And this started a whole project we didn't even know we had, because she figured out that in the acidic medium, so the sour medium, that actually helps the division machinery assemble better and allows the cells to divide when they're short. But in medium that's basic, so less sour, more soapy is how basic is often described in chemistry, although I've never really tasted basic medium, so I can't say for sure. But in basic medium, the division machinery doesn't assemble quite as well, and the cells are longer. So while she was doing this, and again, this came out of one control experiment, she also discovered that pH alters the activity of cell wall synthesis enzymes, the enzymes that make the cell wall. And antibiotics called beta-lactam antibiotics, so cefalexin and the cephalosporins are probably the ones you guys have been prescribed. Those target those enzymes, some very specifically. And if those enzymes aren't active, those antibiotics don't target them very well. And what she found was that at neutral pH, she could give them a certain concentration of drug. So I don't have a pointer, so I'm a little... So if you see the cephalexin inhibits cell division, so at neutral pH, at a certain concentration of cephalexin, she sees long filaments because the cells can't divide, the cells get really long. But at pH 5.5, where the cells are still quite happy, there's no change in cell length, really. If she goes up even higher, if she doubles the concentration of cephalexin of the antibiotic, they don't grow at all at pH 7, but they grow fine at pH 5.5. She also tried this for another drug, mesalenam, which actually we don't use in the United States to treat bacterial infections, but is common in Europe to treat urinary tract infections. She saw the same thing. So mesalenam actually inhibits the longitudinal growth, so cells getting longer, so they become round when you treat them with mesalenam. And at pH 7, those round cells at pH 7, they're still what we call rod shaped. They still look like kind of ibuprofen pills or some capsules, right? At the next concentration up, they don't grow at pH 7, but at pH 5, even though they're maybe a little round, they grow quite happily. If you were just looking at bacterial growth just in a flask where we can't see what they look like, you would see no difference. So just that change in pH made a huge difference in the sensitivity to antibiotics. And I think this is actually something we should think about, because your bladder, actually, pH of your urine varies with what you eat. So, for example, it can go all the way from pH 4.5, which is pretty acidic, up to pH 8, which is pretty basic. At that, if you take cranberry juice, which we've all heard, it's actually a myth, is something to help you get rid of urinary tract infections, that actually lowers your bladder pH. So you're actually, if you're taking a cephalosporin, super common drug for urinary tract infections, and you're drinking cranberry juice, you're actually working at cross purposes. So all of this work with lipids and size and the environment got me thinking. And I went to the dentist one day, probably around 2016, 2017, and they handed me this toothpaste. I love the toothpaste samples, and the toothbrushes are like gold from the dentist, right? But the toothpaste had total, and I'm like looking at it, and it said it's antimicrobial, and it prevents gingivitis, which is just a gum infection. And I look at it, and on the back it has this drug called triclosan. And it has a lot of triclosan. So triclosan 0.3% is the concentration, and this is 60,000 times the amount that Corey Westphal, who was a postdoc in my lab at the time, found was needed to keep E. coli from growing. 60,000 times the amount needed to stop it from growing. So we kind of, Corey and I, started thinking about this, because we know that if you add triclosan, it kills bacteria at high levels by breaking their membranes, but at low concentrations, it inhibits their growth by preventing lipid synthesis, so their membrane can't get any bigger. But when you inhibit lipid synthesis, you get this small molecule that my lab has become kind of obsessed with called PPGPP, which actually Sarah Anderson in my lab just had a paper accepted on PPGPP. But PPGPP has actually been implicated in lots of bad things with antibiotics. If PPGPP levels are high, bacteria are protected from antibiotics. So high PPGPP means those cells, you can give them a bunch of antibiotics, they don't grow, but they don't die. And then you take away the antibiotics and they grow again. So the idea is that triclosan might be protecting the cells from the antibiotics. And in fact, this is exactly what Corey found. We saw a 10,000-fold reduction in the efficiency of a drug called ciprofloxacin, which you guys, I'm guessing, have heard of. Cipro is a very common, if your doctor doesn't know what you have, they often will just give you Cipro. And we did the experiments in mice, actually in collaboration with Ana Flores Morales and the Hulkerin lab on the med campus, and we saw a 100-fold reduction in the efficiency of ciprofloxacin to treat urinary tract infections in mice. So these are kind of the work that we've been doing over the past 15 years. The rules in my lab are pretty simple. Do the right controls. You just saw from Lizzie's work, the advantage of got 3 papers out of control experiments. Always look at your cells. It's a little bit of a dirty secret, but microbiologists often do not look at the cells. On the med campus, in the micro department, I think there are only 2 or 3 labs that have real high-powered microscopes. That's not unusual. And in our department, I think I'm the only micro lab with a microscope that's, we use regularly. And expect the unexpected and be kind. Those are the rules in the lab. The reason we always look at the cells, and I think I've given you some suggestion of that, is sometimes, and expect the unexpected, and this is actually work of Sarah Beagle from my lab. This is Klebsiella, which again is a pathogen. So she treated them with mesalinum, and we expected them to eventually die. This is at neutral pH, and we thought, oh, they're often they're very close to E. coli genetically, but we didn't see that. We saw something really scary. So, sorry, you're just gonna have to watch bacteria grow. They get round, which we expect. They develop these vacuoles, which Sarah figured out are actually coming from this double layer of the membrane. They're full of what we call the periplasm, and they just keep growing. You grow them, and if you look at a microtiter plate or a test tube, they grow just fine. And they're in 500 micrograms per mil mesalinum. It's almost enough for it to start crashing out of solution. It's way above what would kill E. coli. So that's scary, and Sarah's working to understand that. This is Adrienne Brower, who's a grad student in my lab. She had these mutants the other day that when she stars them, they form these sort of vacuoles inside them, and then they explode. We have no idea why. I'm just showing you some of the stuff we see, and it's because we're looking at them. We would never know this if we didn't look at them. Jesse Bullock in my lab. So FTSD is this protein that forms a ring where the cells divide, and it should form a nice even ring right down the middle of the cell, just right where it's going to divide, and that ring looks like it constricts when the cell divides. But Jesse's made mutations in FTSD to understand how it assembles, and her mutants form spirals in the cell. These are proteins, and the cells sort of twist sometimes, and they don't divide very well, and they're very long. This, by the way, is Bacillus subtilis. I know you can't tell, but it's not E. coli. And then Sarah Anderson, we used to have 3 Sarahs in the lab. We're down to 2 Sarahs. But it's just not good to be a bacterium in the Levin lab. So these are mutants that can't make PPGPP. And when you get rid of it, the cells are like random sizes. And the green, this is E. coli, is the FTSD ring. It's supposed to be in the middle of the cell. And it's just not very good. And we get these long cells all over the place. It really isn't good to be a bacterium in my lab at all. This is Ryan Valdez. He's looking at this outer layer on the bacteria called lipopolysaccharide, and all gram-negative bacteria, E. coli, Klebsiella, Neisseria, all have what we call lipopolysaccharide, and its role has been a little mysterious. So Ryan's been found that, along with work from Casey Wong, one of my collaborators at Stanford, that it's probably there to help maintain the rigidity of the cell envelope, and it does it by interactions between magnesium ions, which are positively charged, and the negatively charged LPS molecules. So this is when he gets rid of LPS using a drug, and he grows them in medium that's low osmolarity, so it's tough for the cell to maintain its internal. And so what you're going to see is they're going to grow, they're going to chain a little bit, and when they turn pink, they're dead. So they eventually all go pink, pretty much. So it's really not good to be a bacteria in the lab. In any case, I'm just giving you a taste of our work without, again, no quiz. Don't have to remember anything. I do want to emphasize kind of the sort of things in my lab that are really important to me and that I really try to instill in my lab, that science doesn't happen in a vacuum. I'm not a believer in the word genius. Maybe Einstein, I guess, like he could be a genius. But generally, I think we're better when we work together and we're better when we collaborate. And I've done a lot of collaboration with I have a lot of wonderful collaborators. Rohit Papu's on here. He's here at WashU. We're collaborating with, and then Min Kyung, sorry, I can't point with my pointer. Gary Patty and Kevin Cho are also here at WashU. The woman in the white coat in the middle row is my really close colleague at Wisconsin, Jade Wong, and then next to her in the middle is Suk Jun, another collaborator, and Casey Wong. Just so many wonderful collaborations, and it's great because I don't know, my physics is basically very dated at this point, and I don't remember a lot of my math, but Casey and Suk Jun and also Shri at the top are physicists, and so it's really wonderful to be able to collaborate with them. I've been really blessed. I've had great mentors. Marcia Altschuler is the woman standing in the back behind two students. She was my first professor at Williams College. She taught my introductory biology class as a freshman, which almost killed me. But I learned to think of biology as a language, and that's why I tell my students, you're just learning the language. It's hard, but you need to know these words to understand what comes next. She taught me in a really fun lab on developmental biology that you could answer questions by designing your own experiment, and that was really thrilling. Chip Lovett was my research advisor. He is a chemistry professor, and to the nth degree, he has a stereotype of a college chemistry professor. He likes to blow things up, do demonstrations, he plays guitar. He taught me, though, that failed experiments, which pretty much happened my entire senior year as I tried to write a thesis, can teach something important, and that great teachers can make anything interesting, even organic chemistry. Sorry, Sophia. But my PhD advisor, I really lucked out. I thought I was going to study frogs, but my first rotation was in Rich Losick's lab studying bacteria. And I found this picture of Rich, and I really love it because this is how he is. Sometimes he twists, Barbara knows, because Barbara Kunkel also had him as her PhD advisor. He twists his eyebrows when he gets really excited. But he taught me to be passionate about what you do. Study something you're excited about. He also taught me that communication is really important. We would go through talks 3-4 times before he let us give them. He taught me, and I tell my undergrads and my grad students this, if you don't understand a talk, it is not your fault. It is on the speaker to communicate clearly. He taught me that teaching and research are synergistic. I was talking to Jonathan the other day. Rich teaches a giant class. Actually, he's now retired, but he used to teach Biochem 10, which was a few hundred students. He taught that class. He's a National Academy member, and he taught it, and it was amazing. And he complained about all the other faculty and how they weren't as good. So. And he taught me that being a great cheerleader is integral to being a great PI. And then my postdoc advisor is a lot more chill, Alan Grossman. He taught me that we don't hide things. We don't not talk about our research. We talk about it. We're not doing experiments in secret. And he taught me to follow the data even if it takes you to unexpected places. The classic meeting with Alan would be showing him a plate on which there are 100 bacterial colonies and one looks a little funny, and he's like, what's wrong with that one? And you're like, oh, please. And he also taught me to play nice, be nice, it comes back. And both of them taught me that the best science happens when you surround yourself with smart, enthusiastic people and let them do their thing. Guide them, but don't tell them exactly what to do all the time. Have a conversation. Even if they're new in the lab, your job is to really help them kind of become their own independent scientists. And they will have ideas. Many of the experiments that I just told you about were not my idea. They were ideas my students had, and I might have helped them, or postdocs had, helped them come to the right experiment, but I did not have a list of 20 experiments and tell them to do those experiments. I continue to be really blessed. I feel really lucky to be here at WashU. I'm really happy that Ralph is here. He hired me. He's still the chair to which all other chairs have to measure up. And I'm sorry, Ram isn't, it may be good Ram isn't here. Ram is doing a great job, by the way, but that's a very high bar. Ursula Goodenough, when I first had Isabel, who's in the front row, I was like nervous. I had to take her back and forth. She wouldn't take a bottle. Sorry, Izzy. I had to drive to daycare. Anyways, Ursula's like, I had 5 kids and I breastfed them all. And I was like, okay, this can be done. Jim Skeeth was my friend who's in the genetics department, and he really helped me learn how to write a good grant. I had no idea. He really helped me. Bob Kranz was my faculty mentor, and he never steered me wrong, never once. Always kept me on the straight and narrow. Barbara Kunkel, I didn't overlap with her in Rich's lab, but she's the older sister of one of my friends from grad school, Maya, and it was just really nice to have her here. And she lives in my neighborhood, and we have some really wonderful discussions. My friends, Hanny and Ram and Joe, and then Liz Malott, who's my faculty mentee, but I get to co-teach micro with her, and it's just been so fun to teach with somebody who's new to teaching this particular class she's taught before, and it's just been really fun to bounce ideas off her. I also want to thank Susie Stark, my mom friend who's here. Our daughters have the same birthday and met at daycare, and that's been a really great friend for me. And then this elderly gentleman standing next to me is Elio Schechter, and his work, he was the first really to do quantitative microbiology on bacteria, on E. coli, and really try to think about what's going on. Can we quantify how big they are? Can we quantify how fast they're growing? Can we quantify what's inside those cells? And his paper in 1958 kind of is like a North Star for my lab in many ways. We keep going back to it. And then obviously my lab. The one in the middle is sort of the first picture of the lab. We've tried to take one every year. I think we're missing 2012. It's really just been a joy to come to work every day. It's just, I love it. Sorry. And I can't thank you guys enough. And then I do want to just end by saying one of the things, and this was mentioned by Jonathan and Dean Hu, that I'm really passionate about, and one of the things I believe is that doing research is something everybody can do. Basically, nobody should be afraid of doing research. I always tell students, and sometimes then they're like, I can't do it. But if you can bake brownies from a mix, you should be able to do molecular biology. Honestly, you just have to measure. The thinking part takes time, but actually doing research is not that hard. And so I want to bring students in, and WashU's made this big effort. I think we're at 20% students who are Pell Grant eligible, basically on full scholarship here now, and first generation. And that combination of being a first generation student and on scholarship seems to be those students need the most kind of, not hand-holding, but they need the most kind of like, here is a place for you. And so I've worked really hard to get this program started. It's still very small. And it really is to bring in these students and to put them in a lab early, because my lab as a grad student, even as an undergrad, was kind of home base. I could leave my backpack there, I could hang out there, I could talk to my PI. And I want that to be that for these students. And so Enter is basically open to any first-year student who meet these 3 criteria. They cannot have research experience. I want no one who has research experience. If they're gonna learn bad habits, they're gonna learn it from my lab. Students select a lab. I poll for PIs, and they commit to the lab for 4 years, but they don't have to stay. We often have them say, I have had 2 or 3 in my lab who are like, I really want to do neuroscience. I'm like, we just had a pandemic. But no, I don't say that. I let them do neuroscience. The PIs are the ones who have to make the commitment for 4 years. And they guarantee 10 to 12 hours a week, and they pay them. This is really important. We pay them $15 an hour, and they're guaranteed pay for doing research in the lab. And in my lab, and I think a lot of labs, they start by doing glassware and media prep. Izzy can tell you, and Sophie too, that first year is really confusing. So having something to do where you're just washing glassware or making media, it's fine. You don't need a big research project. And then you transition, and Nina's transitioned with Ryan as her mentor to doing research. And then the idea is that, and I'm sure Nina, who's only a sophomore right now, will have plenty by her senior year to write a thesis, but that's the hope. So thank you very much for your attention.
I just told Petra that no one's allowed to ask her questions today. Thank you, Petra, for the truly fascinating presentation. I am a plant biologist, and I think plants are really cool. Had I had a professor like Petra, I might have become a very good microbiologist, competing for grants with you. So thank you all for joining us today, and please join us for the reception in the back of the room there.